So blood glucose may be derived directly from the following sources. Number one is when we have diet, when we have food, the carbohydrates get digested into the most basic unit of glucose. When the digestion is complete, the intestinal lumen cells absorb glucose from the intestinal lumen into the cell, and from there it will transport it to the portal circulation. That is one way how normally whenever we are having diet or when we are having food, glucose enters the bloodstream. Second is when in a state where we are in a state of fasting, that is around 3 hours, 2.5 to 3 hours of having food, our body starts to utilize glucose based on the storage, how much storage we have. So blood glucose is stored whenever the body does not require it, or whenever there is increased concentration of blood glucose, it is stored as glycogen either in liver. First of all, in liver, when the space in liver is complete, it will be stored in muscles. Okay. So first step is whenever our body requires energy after a prolonged fasting state, it is breakdown of glycogen that is stored in liver. So this process is called as hepatic glycogenolysis. Okay. So this happens in this sequence only. First enters through absorption. Second, there is hepatic glycogenolysis. Now, when all the hepatic glycogen is completely utilized for production of glucose and our body still requires some amount of energy, so our body undergoes a process in liver called as gluconeogenesis. So that is production of glucose from non-carbohydrate sources. So when all the stored carbohydrate is empty, stored glucose is exhausted, and blood glucose level is becoming low, it is coming down, our body starts to produce glucose non-carbohydrate sources such as glucogenic amino acids, lactates, pyruvates, glycerol, and propanil. So overview of this, we can say whenever our body requires glucose and all the storage is empty, our body undergoes a process where glucose will be produced from proteins, amino acids, from lipids, okay, all other macromolecules that are present that will be utilized for production of glucose, and this process is called as gluconeogenesis. Now, glucose can also be obtained from other carbohydrates such as fructose, galactose, etc. Uh, Disaccharides and oligosaccharides that are present in the body can be broken down to produce glucose, and that is the last step or the last factor or last option for glucose to get entered into the bloodstream. So this happens in this sequence. First happens to the absorption from the intestine. Next there is hepatic glycogenolysis. Then there is hepatic gluconeogenesis, and finally other carbohydrates, breakdown of other carbohydrates such as fructose, galactose, and other oligosaccharides to release glucose into the blood. Yes, next is rate of removal from the bloodstream. How the glucose will be removed from the bloodstream. Number one is oxidation of glucose by tissues to supply energy. So whenever our body requires energy, circulatory glucose which is in the bloodstream will be taken up by the cells. Okay, so this cells will oxidize glucose and produce energy. So this process is called as glycolysis. So breakdown of glucose to produce energy is one way of removing glucose from the bloodstream. How does it do it? Whenever there is requirement of energy, circulating glucose will be taken up by the cells and will be undergoing a process called as glycolysis. Next is whenever our body does not require any energy and there is large amount of glucose circulating in the blood. So our body thinks that maybe on the later time our body might require energy, so it will store glucose. Okay, so storage form of glucose is glycogen. So glycogen's formation starts to happen, mainly starts with liver. When liver is completely full with glycogen stores, it will go into muscles. So this process of formation of glycogen from glucose is called as glycogenesis. So first hepatic glycogenesis will happen, then muscular glycogenesis will happen. It's one of the ways to remove the glucose from the circulation and store it as glycogen. even that is empty. Okay, when that there is no more space available and still there is large amount of glucose circulating in the blood, next thing happen is called as lipogenesis. So lipogenesis is a process where lipids and fatty acids are produced, synthesized from glucose. Okay, so this happens in the adipose tissue where glucose is utilized for production of lipids and fatty acids. So we can correlate this. If you have or eat sugary foods for a long period of time, we start to gain weight and become fat. Okay, so that is the reason. So whenever there is complete storage of glycogen and still there is some amount of glucose circulating in the blood, it will be used for production of lipids and fats in adipose tissue and there it is stored. Next is synthesis of compounds containing carbohydrates. Okay, so once that is also over, lipogenesis is over, but still there is amount of certain amount of glucose is still circulating in the body, which is above the normal range. So it undergoes a process where uh, compounds containing carbohydrate containing compounds are synthesized. For example, fructose, lactose, glycoproteins, and glycolipids. So it will be used for production of oligosaccharides, derived lipids, derived proteins, also formation of ribose sugar for glucose required for nucleic acids for DNA and RNA formation. And even if that is not possible and still there is a high concentration, our body removes that glucose through urine in a process called as glycosuria. So glycosuria is presence of glucose in urine. So still after all these things, after using it for energy, after storing for later use, after producing lipids and fats, and after synthesizing derived carbohydrate compounds, still it is present. It is still the concentration of glucose is higher in blood. This leads to formation of glycosuria, but body simply removes it from the circulation by excreting it in urine. So there is a threshold for it. Certain concentration of blood glucose should exceed for this. We'll see it later. But it is also one of the mechanism by which glucose is removed from the blood circulation. Right. So now the mechanism is very important. Okay. So how our body actually regulates or controls the blood glucose within the normal range. First of all, let us understand the normal blood glucose concentration throughout the day, irrespective of at what time the person had food or not, should be between 70 to 140 milligram per DL. Whenever 70 decreases less than 70, there are mechanisms that are present that will start increasing the blood glucose level. Similarly, if it goes beyond 140, there are mechanisms that are present within our body which will decrease the concentration through different processes. Okay, let us start with the first one. Let us say our body is in fasting state. So what is fasting state? Is a state where there has been 2.5 hours to 3 hours from having food, or whatever food we had through our diet is completely digested, absorbed, and either utilized for production of energy or all the glucose is stored. So when our body enters this fasting state, the blood glucose level starts to decrease. Okay, so when it goes beyond 70 milligram per DL, which is indicated by a decrease in blood glucose level, it acts as a stimulant for alpha cells of islets of Langerhans. So islets of Langerhans is the group of tissues that are present in between pancreas, and these islets of Langerhans are responsible for synthesis of two important hormones related to glucose metabolism. Okay, so there are alpha and beta cells. Alpha cells are responsible for synthesis of an hormone called as glucagon. So glucagon is a hyperglycemic hormone. Its role is to increase blood glucose level in the body. So whenever the blood glucose becomes less or there is a decrease in blood glucose level, our body compensates this by increasing the synthesis of glucagon. So glucagon increases the blood glucose level by the following processes. So it starts to increase the process of breakdown of glycogen called glycogenolysis. It starts to produce glucose from non-carbohydrate sources such as gluconeogenesis. It decreases glucose utilization. Okay, it prevents utilization of glucose because the body requires blood glucose level, and decreases glucose uptake from the cell. Okay, so make sure that blood glucose is circulating between 70 to 140. Let us understand why this maintenance is very important. Our brain, central nervous system relies solely on glucose. So there are no other energy producing molecules that will be utilized by the central nervous system, brain and spinal cord. So it's very important that in CSF, the glucose level is always high. CSF is extension of blood. Okay, it is just like blood for CSF, central nervous system. So our central nervous system can only utilize glucose, so glucose has to be maintained between 70 to 140 milligram per dl. So whenever it decreases below 70, our body undergoes a fasting state or state of fasting. So this decrease in glucose below 70 milligram per dl acts as a stimulant for alpha cells of islets of Langerhans. So alpha cells starts to produce or increase the production of an hyperglycemic hormone called glucagon. So glucagon will increase the blood glucose concentration by increasing breakdown of glycogen, production of glucose from non-carbohydrate sources, decreases glucose uptake by the cell and its utilization in the cell. So this process continues till the blood glucose returns back to this level between 70 to 140 milligram per dl. Okay, so it is going to continue till here. We'll have a time frame when our body is in homeostasis for blood glucose. After some time again, the glucose level starts to decrease, and the mechanism that is there on the left side of your screen will continue. Okay, so that is one stage in the day when we are in a state of fasting. At the same time, when we are in well-fed state, that means we had food. Okay, we had food. We had a nice heavy breakfast. So our blood glucose level is starting to increase. It is increasing more than 140 milligram per dl of blood. So this increase in blood glucose level, when it goes more than 140 milligram per dl, it acts as a stimulant for beta cells of islets of Langerhans. So beta cells present in islets of Langerhans are responsible for production of a hypoglycemic hormone called insulin, which is responsible for decreasing blood glucose level. So whenever the blood glucose is going more than 140 milligram per dl in a well-fed state, our body increases the synthesis of insulin in beta cells of islets of Langerhans. So insulin increases the utilization of glucose and brings blood glucose level back to the range by increased cellular uptake. It forces the entry of glucose into the cell either for production of energy through glycolysis or storage into glycogen through glycogenesis. It also promotes or increases lipogenesis, HMP shunt pathway, which is another breakdown of glucose, and decreases other sources. Okay, decreases gluconeogenesis and glycogenolysis. It promotes mechanisms which will decrease blood glucose level. At the same time, it uh, will not promote the mechanisms which will increase the blood glucose level. So by all these mechanisms, insulin continues its work till the blood glucose comes back to this range, till 70 to 140 milligram per dl. Okay, so once again, when it goes more than that, we can see there is increase in blood glucose level, beta cells are stimulated, insulin is produced, insulin will increase cellular uptake, it will promote glycolysis in the cell. It will promote storage of glycogen. It will promote production of lipids. It will promote HMP shunt pathway and decrease production of glucose from non-carbohydrate sources and breakdown of glycogen, or to maintain the blood glucose level between 70 to 140 milligram per day throughout the day.